got some of the carbon nanotubes from Rick Smalley in 2005, came up here and met John for the first time in face-to-face, August 2005, put these nanotubes in his machine and they heated. Rick Smalley then passed away a few months later. We began the RF testing. John got us a unit down to MD Anderson. We started doing some of the testing. And we actually found that we put these carbon nanotubes in there and we got great heating. We had a paper published on that. We actually then started doing some of the treatment in rabbits. We continued then essentially looking at some of the gold nanoparticles. Had another manuscript published in December using the gold. January had another manuscript, again, this showing that we could target the gold nanoparticles. Started doing more targeted testing summer of 2008. And this is again using some of these new antibodies that I told you about. We started doing work then, as I told you, optimum size, shape, charge, all of this. All of these are things that are ongoing. Just had a paper published on that 2 months ago. December, we had the paper that actually showed that we could use specific types of antibodies to get them taken into the cancer cells, again, proving we could do that. Unfortunately, in February, we lost John. And a huge loss personally, huge loss professionally. But again, I want to emphasize that it's done nothing. If anything, it's actually increased our activity on this. I set my entire lab staff down the week after. I said, look, we're all sad, but we made John a promise. We made Mary Ann a promise, and I'm going to keep my promise. We are going to get this to clinical trials. We are going to assure that this treatment becomes a reality. About a month after John died, a patent was approved. Important thing, showing that John's idea really was novel. And this is where we're at now. The work is continuing. We're continuing to push forward. We're continuing to move this very rapidly. And here's just an example of what I'm talking about. Again, here's John with a mock-up of what a human-sized device might look like. Again, a couch sort of like a CT scanner where you'd move. We're not telling fish stories here, I assure you. Here's a mock patient. Mary Ann, thanks for being a volunteer. I think she was asking, there's not really anything up there in that box. And here I am with John and the Rutkowskis, talking about this. And again, this has been a great collaboration, a great partnership with the Erie community, for me, with John, with the Rutkowskis, with many other people up here. Again, we can talk on the fly. I mean, this is stuff that's happening in real time. We're changing designs, we're talking about ideas, we're getting people involved. Again, Charlie Rutkowski, and once again, John and I there at Industrial Sales, and just talking about this and some of what we're doing, where we're heading with this. So the long and short of it is we are continuing to make progress. We've got a number of manuscripts that are now submitted. These are things that I've shared bits and pieces quietly with you. Again, I wouldn't do that with a scientific audience because, again, of all the rules. We've got lots of rules in academia. You can't discuss results until they're officially accepted. But we've continued to work at a higher and a faster pace. We've been working with a number of types of cancer, working with a number of types of new molecules, and this work is proceeding. And John's legacy will survive and it will be intact. And so, again, this is a quote that I made in 60 minutes, and I stand by it. It hasn't changed. This is the most exciting thing I've seen. It has some of the greatest potential I've ever seen as an effective, novel cancer treatment with the potential to be minimally toxic, and that's a key. And with that, I will assure you, those of you who've never visited Houston, this is not lovely Galveston Bay. We don't have any volcanoes in Galveston. This is Naples, Italy, where I also do some liver research. And with that, I will say thank you, and I will be happy to answer any questions.
When these antibodies attach to the receptor cell, are they always, or to the receptor site, are they always taken into the cell then? Is that just a natural process, or do sometimes they sit on the outside? Or
the answer is, it's a long answer. Usually, yes, it's a very rapid. As soon as you get binding to the receptor, you get rapid internalization of the receptor. There are a couple of exceptions with different types of receptors that literally sort of spit off what bind to them. But again, we're binding to it in a fashion that it won't release it. So those receptors are, yes, very rapidly internalized. There are ways you can alter that. We don't want to alter that. We want them internalized. We want them taken in. We have yet, we actually are getting even fancier yet. We're putting a second targeting molecule on them after they're taken into the cell. Right now, we're very good at getting them into the outside of the cell, the cytoplasm it's called. We'd love to get them into the nucleus, because if we can destroy the DNA, we can kill the cells even faster. So we're adding actually now a second derivative targeting molecule. So as soon as they're taken into the cell, then that second molecule is activated and they're taken up into the nucleus. So that's the thing we're doing now with some of these to get even fancier. Sir?
Two questions. One, when do you expect the human trials to begin based on the current pace of things? And then secondly, mortality rate, is that what's going to be used in determining which cancers are targeted next, such as brain or ovarian?
The answer is I don't have an exact answer to your first question. I'll know a lot more after I meet with the FDA. We believe we're on pace to be in human clinical trials potentially as early as 2 to 3 years. But they could always add on a few additional tests or things they want to do. I've already had some conversations. I'm actually already doing another study that they specifically requested on another type of phenomenon that may improve this heating. So they could come to me and say, we want you to do the following things, and I could add 6, 12 months on it. So we think we're within a few years is the easy answer. The answer is after we get approved to treat, say, pancreas or liver cancer, we could then open the floodgates and start treating any kind of cancer we can target. And I've got colleagues who are interested in targeting glioblastoma, very much in the news right now since Senator Kennedy just passed away from that cancer. That's a very bad cancer that we don't have good treatments for. We are looking at breast, lung, prostate. So the answer is we're going to quickly open it up to others. We're continuing to do studies on those, even though clinically we're going to focus on those first 2. Yes, sir.
What prevents you in terminal cases of jump-starting clinical trials, human clinical trials?
There used to be a mechanism through the FDA and through the NIH where you could put in what's called a compassionate IND for somebody who's really failed everything else. The problem is what they quickly realize is with most new treatments, if you take a patient who literally is kind of tried everything and may have a survival measure only in weeks or months, if your treatment, say, works but not well enough and that individual still dies, then they come back to you and say, gee, this maybe it helped, maybe it didn't, but this isn't sufficient. And God help you if the patient actually dies very rapidly, they say, well, it looks like your treatment killed him. So, and that may have had nothing to do with it. So that mechanism now is used very infrequently for that reason, because there have been a couple of very good drugs that actually got essentially derailed completely because of that and had to then go through several more years of extensive testing. So nobody is really too fond of that measure or technique anymore. Again, unless I get data from my laboratory that really says we think we can hit a home run with this, it would be dangerous to use that technique. Sir?
Tell us about your funding. Where do you get your funding now, and what do you project your funding needs for the next year or so?
Our funding now comes from philanthropic sources.